do a lot of traveling.
I'm glad you warned us because I'm not going to talk now.
Oh, thank you. All questions will be given in writing, and I'll have it back to you by Thursday.
Okay.
Okay, so.
Can we turn at least one light on? Sorry, do you mind? Just so I don't fall asleep when such a rain.
I know, it's rough. Okay. So I'm just going to, I've got his thing, we're just going to start playing with it, going through it. So we'll start with 17, urinary system. Here's your urinary system. Glomerulus, glomerular capsule. Peritubular capillaries carry the blood, carry what's been reabsorbed. Now, you need to make sure that you differentiate between absorption and reabsorption. Absorption does not happen in the kidneys. Absorption happens in the intestines. Because the pressure of the afferent arteriole, the pressure that the blood is pushed into Bowman's capsule, essentially indicates how much filtrate is going to be formed. The higher the pressure, the more filtrate is going to be formed. And then you go through the reabsorption. Okay? Sorry, I've got two different things. I do not think this will work with this. Okay, so you have glomerulus. You know, this is really the nephron. You got the glomerulus, you got Bowman's capsule, you got the proximal convoluted tubule, descending loop of Henle, ascending loop of Henle, distal convoluted tubule, and then the collecting duct. Okay? So if you'd ask how the fluid is going to go through, that's the pathway. And you have your peritubular capillaries, and you've got your peritubular capillaries vasa recta. They're picking up the nutrients. They're picking, the big thing is the water that comes out that's drawn out by the salt is picked up in the peritubular capillaries and taken back, dumped into the vena cava, back into the system. You know, what's happening here with the countercurrent multiplier that goes down and the salt concentration on the outside gets higher at the bottom because of the pumps, is you've got more water being pulled out. They are picked up by these capillaries and whisked out of it. Then as you ascend, where there is less gradient, or actually maybe a higher gradient back into the ascending loop of Henle, it's impervious to water. You're pumping salt out, but it's impervious to water. The water cannot get back in. Okay. Regulation. Functions of the kidney. Regulation of extracellular fluid, volume of plasma, blood pressure, waste, electrolytes, pH. And if you remember the quiz, that's all of the above. Urine is made from the kidneys, pools in the renal pelvis, down the urinary bladder. Passes from the bladder into the urethra, transported using peristalsis. What's peristalsis?
Everything moving of stuff through a tube.
I'm not sure he will accept that.
They're
Contractions. Muscular contractions, timed muscular contractions that will move either fluid or solid through a tube, i.e. The intestines, chyme moving into the small intestine, feces moving out of the large intestine through the rectum and the anus.
What about the food coming down your esophagus?
Yes, absolutely. That's a peristaltic action. You guys brought me Cokes, right? Thanks, but I got some. No, thank you. Okay, we started off going over the function, the structure of the kidney very quickly. You've got glomerulus, Bowman's capsule, proximal convoluted tubule, descending loop of Henle. Through, and then down, descending loop of Henle, you get the bottom, and then the ascending loop of Henle, then distal convoluted tubule, collecting duct, and then your ureter. Kidney functions, and I'm just kind of looking at this. Nephron structure, urinary system, regulation of cellular fluid, blood plasma, blood pressure, waste, BUN, things like that, creatinine, electrolytes, it's excreting or reabsorbing, it helps maintain electrolyte balance, and pH with the excretion of hydrogen.
So regulating fluid and waste. Right?
Yes. Okay, urine. I'm sorry, I've got one that does the laser and one that does the slides. Okay, here it is in the body. There's your kidneys. The kidneys are at the end of the chain. There's no collateral circulation to the kidneys. Even in your heart, you get a vein that's blocked, you've got collateral circulation that still works to feed the heart. The kidneys are at the end of the line. There ain't no collateral circulation. If that breaks down, the kidney goes. That's why you've got two of them. You've got a backup. So you've got inferior vena cava, that's taking blood back up to the heart. You've got the kidneys, renal vein, renal artery, abdominal aorta, ureter, urinary bladder, urethra. Kidney has two distinct regions. You've got the cortex and the medulla. Each region drains a minor calyx, major calyx into the renal pelvis. The minor calyx is your nephron. It drains into the major ones and into the renal pelvis. Micturition, urination. Is he going to get into this?
No, skip.
Okay, you need to tell me what he's not going to do, because I just want to skip those slides, because like I was telling everybody else, I got 500 slides here.
Okay.
And I need to know what to skip.
Okay.
Do you have this like clip of the questions that
I have it.
Oh, okay.
Yeah, I have it. Thank you. A microscopic nephron functional unit. More than 1 million of them. Small tubules and associated blood vessels. What's your tubules? Loop of Henle, ascending, descending. Collecting duct. That's in every nephron. You got 1 million of them. We just went through this. Renal artery we talked about. Interlobular. These are all.
Keep going.
Skip.
I realize that. Nephron. Glomerular. Bowman's capsule surrounds the glomerulus. Make up the renal corpuscle. Filtrates produced there. Filtrate. It's pushed through. The stuff is filtered out. There's big stuff that doesn't get through, such as proteins, albumin. Amino acids in that, singular, will get through. But big proteins, like albumins in that, will not get through. Because in your distal convoluted tubule, you're reabsorbing things like that. We'll get to that. Okay, next fluid pass, descending and ascending loop of Henle. Filtrate produced in the renal corpuscle, passes into the proximal convoluted tubule. I could see him saying, trace a drop of urine.
Yeah.
Trace a drop of urine. He's kind of said he's going to do it with food when you get into the digestive system. Yeah.
Glucose gets reabsorbed, right?
Glucose gets reabsorbed.
So it gets filtered.
Yes. Yeah, it's too small. They can't not filter it.
Why is it again that when you have an increase in stress that the glucose goes into the urine and gets pretty good?
Oh, that was just a story I made up.
Big, small.
Because stress in your flight and sight, Flight and fight syndrome. Your body releases cortical and mineral steroids. And mineral steroids cause your blood glucose to go up because your body says, whoops, I'm going to need glucose, I'm going to need insulin, I'm going to break down glucagon, I'm getting ready to haul out of here or fight somebody. It shuts off the blood to the kidney. The circulation to the kidney under fight or flight goes way down because it's pushing the blood to carry nutrients to the heart, the brain, and the muscles. Okay? But it's the release of the mineralocorticoids and the steroids that cause the blood sugar to go up as part of the fight and flight. Okay? Okay. Loop of Henle passes into the distal convoluted tubule, collecting duct. Now urine will drain into the minor calyx. We've seen this one before. Two types of nephrons, juxtamedular, better at making concentrated urine up here. The cortical is down here.
Keep going.
Glomerular filtration, capillaries are fenestrated, large pores.
Yes.
Okay? Called filtrate. We've been there. Must pass through the fenestrate. Glomerular basement membrane, visceral layer of glomeruli. I don't think he's going to get into this.
Skip.
Corpuscles, efferent arterioles, they're carrying the blood in. That's what controls the amount of filtrate, the pressure in those. And it's pushed out.
Pressure to an efferent artery?
In the arterioles coming in, yes. Because the pressure is going to, because you got in there, it's fenestrated, so it's able to push fluid out. It's what it's designed to do. That's where the fluid comes from. Glomerular corpuscle, slits in the pedicles called slit diaphragm, major barrier to the filtration of plasma proteins. Defect here, proteinuria. Albumin is filtered out but is reabsorbed. Now, there may be a few things that he's going to talk about. Know the difference between sympathetic and parasympathetic. Have an idea of endocytosis, exocytosis, though I'm not sure that he got into that on the thing. Filtrate. Glomerular capsule gets there via hydrostatic pressure of the blood. The blood pressure pushes it through. Colloid osmotic pressure, I don't think is really very much. Very permeable capillaries. Filtration rate. That's all the glomeruli, all the nephrons in both kidneys in 1 minute. 115 to 125 mils. Regulation. Vasoconstriction of dilation of afferent arterioles. Again, extrinsic system, sympathetic nervous system, controls the amount of blood that goes to the kidney. In fight or flight, you don't want your blood going to the kidney. You want to carry nutrients to the heart and the muscles.
Wait, can you go back?
No.
Thank you. Okay, thank you.
Okay? Sympathetic nerve effects. Fight and flight reaction, vasoconstriction of the afferent arterioles. Okay, we got that? That's just what I went over. Sympathetic nerve effects, blood pressure, baroreceptors, exercise, increased cardiac output. We're going on. Reabsorption of salt and water, 180 mils fluid, 180 milliliters of water. That's wrong. It's 180 liters of water is filtered per day. Only 1 to 2 liters is excreted as urine. This will increase when you're well hydrated. Minimum of 400. Obligatory water loss. You've got to have that much to get the waste out. You've got to have that much to get the waste out. That's obligatory water loss. 85% occurs in the proximal tubules and descending loop of Henle. That's unregulated.
Is that in a 24-hour period, 400 mL?
Yes. Yes. Reabsorption in the proximal tubule. The osmolality of the filtrate in the glomerular capsule is equal to that. When it goes out, if the osmolality is equal, what is it? Isotonic. Okay? Sodium is actively transported out of the filtrate into the paratubular blood to set up a concentration gradient. So, let's see. What's happening here is, let me go back to this one. As you're coming down here, here's your loop of Henle. What happens is there are active salt transport. It is pumping salt out of the urine, or out of the filtrate, into these spaces. It creates an osmotic gradient that's going to draw the water out. You use probably 15 to 20% of your body's energy of the food that you take in to do that. It takes a lot of energy. It's going on all the time. It's pushing that salt out. It uses 1 ATP each cycle, and then it draws the water out. The water then, as I said, is picked up in the paratubular capillaries, back into the vena cava, back into circulation. As this moves down, this gradient becomes more and more and more, so it's drawing more water out. So by the time you get down here, you're at about a minus 1,400 milliosmoles. As long as that's permeable to water, it's going to keep sucking it out. Now the question on the lab was, what about animals in a dry environment? Their loop of Henle is much longer, which allows that gradient to get much more, so by the time that urine is done, it's essentially solid.
We had a beardy, and they had
You had a beer?
a beardy dragon.
Oh, okay.
And so when they go to the bathroom, there's a white capsule and a brown capsule. They're like solid.
Yeah.
Like a solid pee and a solid poop.
Right. Yeah, that's good reabsorption.
So, Mrs. Smith, so it goes from
Sorry.
osmotic to osmolality, right? Because of what's happening?
No,
Oh, no?
it's all osmolality.
Okay.
In the descending loop of Henle, you're pumping salt out. You actively pump the salt out. That creates the osmotic gradient, and as we showed in the lab with the egg, we kind of drew the water out. The osmotic gradient is to push it back in. We drew the water out with TerraServe. So we created this very concentrated external environment. This is exactly what the kidney does. It does it, however, by pumping salt out. And you know how the egg shrunk. All the water came out of the egg. The same idea would be in concentrating the urine. If that was the descending loop of Henle, that water that came out of the egg, instead of just going into the TerraServe, would have been picked up by the paratubular capillaries and put back into circulation. But that's how that was supposed to work. And it worked well. Okay, nephron. Let's go back. I'm going the wrong way. Regulation of filtering, sympathetic, reabsorption. Okay, this is liters, not milliliters. Increases when you're well hydrated. You feel a lot more when you're well hydrated. With the exception of the times if you're drinking a lot of beer or drinking a lot of caffeine. Getting a lot of caffeine, because those affect the ability to reabsorb water. And I think we'll touch on those, though we may just skip it. Reabsorption in the proximal tubule. Osmolality of filtrated glomerular capsule is equal. So when it's filtered, it's equal. As it moves down the proximal tubule, the sodium is pumped out. It creates the osmotic gradient like we talked about. Active transport. Cells of the proximal tubule, tight junctions on the apical side facing the inside. On the apical side also contains microvia. These cells have lower concentrations than the filtrate inside the tubule due to the sodium-potassium pumps. On the basal side, sodium from the ultrafiltrate diffuses into these cells and is pumped out the other side. So you're actively pumping sodium out. That's the take-home message.
In the ascending.
In the descending.
Wait,
Did I say ascending?
why would you pump sodium out on the descending?
Because that creates your osmotic gradient.
But on the descending,
It's
it's
ascending.
permeable to water, not sodium.
I know. You pump the sodium out, it creates the gradient outside the descending loop. It draws water from inside the loop out.
But the sodium's being pumped at the ascending.
It's pumped on both of them.
Oh, wow.
Sodium is pumped out on both of them.
And we're just gonna focus on the one he told us.
Keep going.
We understand, because it happens all over, there's secretion in the proximal, you know, so
I think we have we'll look at this.
we believe you, we're just not adding that to our
Is he
life.
saying sodium isn't pumped out on the descending?
You really wanted us to know that sodium, that it's not permeable, it's only permeable to water on the descending loop, and only permeable to sodium on the ascending loop.
You need to see
Give us
this.
the calcium shows in the book.
Am I
You're
saying,
talking
maybe I
about what she said
misunderstood
in
it
the
as a lecture.
Yeah.
The concentration goes down?
Like if a baby was going to the bathroom for the first time, there would be no gradient because it hadn't reached the ascending loop yet.
Like,
Right?
what number?
52. For the decent.
Yeah.
Pumps out water and the decent pumps out filtration.
Let's go on. We will.
Okay,
We'll come back to it.
Ascending, absolutely. You're pumping sodium out.
and that's happening first, so then that makes it
No.
salty.
No, that happens second. The sodium pumped out of the ascending is still drawing water out of the descending.
I just got more confused from my side.
Okay, let's go on. We'll get to that.
That's not true.
Pumping of sodium into the interstitial space attracts the chloride. The water follows that out into the interstitial space. You got that?
And
Yeah.
the descending, yes.
Water increased concentration of salts and water diffuses into the paratubular capillaries, which the increased salts again draw more water out. Proximal tubule fluid, reduced by a third but still isotonic, isomotic. Plasma membranes freely permeable to water and salts. That's in the proximal tubule. Descending loop of Henle. 20% of the water is reabsorbed. It happens continuously, is unregulated. Final 15% is absorbed later in the nephron under hormonal control. Fluid entering the loop of Henle is isotonic to the extracellular. At the top, it's isotonic. At the bottom, it becomes more concentrated because of the sodium that's been pumped out. Descending loop of Henle. Not permeable to salt, but is permeable to water.
That's where.
Okay, it's not permeable, but it's by active transport. The salt is out.
Right.
It makes
It's not
sense.
diffusing out, it's going out by active transport.
And I don't want to confuse anybody, but maybe, so in the descending, there's water's moving by osmosis, right?
Right.
Yes, yes.
The ascending is getting actively,
No.
it's just falling to sodium.
All in the ascending, it's becoming more dilute because the sodium that's left in there is being actively pumped out.
Okay, so in SI, what they
And
were
it's
saying
falling,
to us,
water
this is
falling.
where my confusion is coming in, is that the first thing that happens is sodium is pumped out into the
Interstitial
into
space.
the interstitial space. And that creates the gradient to allow diffusion of water to happen in the descending loop. That's what we were just taught.
That's fine.
Did I hear that wrong?
That's fine, you're good with that.
Okay,
You're good with that.
alright,
I'm good with that,
okay.
okay?
Can we stay a little bit back on that slide for a minute? I just want to invite a couple of things.
Don't you have a camera in that little thing?
No, it's just I can't, you know.
So sodium is still happening everywhere.
Yeah. And I think in the thing you showed me, Rose, sodium was going out everywhere too.
No, it's
No.
going out.
No,
It's
so
going out.
it's going out in the ascending end.
Okay, we'll clarify that as we go.
Okay.
This is right out of your book.
Okay.
This is right out of your book.
Got it.
Okay. Countercurrent multiplier. Water cannot be actively...
Skip,
Say what?
We don't need this one.
I know.
He's not going into this?
Solid
He
concentration.
said it doesn't even know, the word doesn't matter.
Okay. This would match what your ascending portion sets up the gradient. That's where it's pumped out.
Okay.
Salt is actively pumped in the ascending loop. Salt is actively pumped into the interstitial space. Moving the sodium down its electrochemical gradients, pulls out chloride and potassium, moves into the interstitial space, water, chloride follows it passively, and so you're pumping out solids in the ascending loop. The ascending loop is impervious to water. Okay?
You know, Mr. Smith, he did say those are water and sodium chloride are some, but just think there's more than just that being reabsorbed.
We'll get into the
No, but
reabsorption.
he did say that. I'm looking at that. He did say that. Those are just some, but there's more.
Yeah, there's a lot of stuff.
He says there's a lot going on. Just those are just some. I showed
But
you.
this gives you the concentration
Yes.
gradient
Yes.
for getting the fluid back.
Yes.
And, the ADH, that's the hormone that's.
We'll get to that.
Okay.
Almost there.
Ascending loop. See right here, you've got some sodium that goes back and forth here. You're using this ATP to ADP to pump sodium out. You have potassium and chloride that will follow it. The ascending loop. Walls are not permeable to water. You have no osmosis from there. Surrounding interstitial fluid becomes increasingly solute concentrated at the bottom of the tube. Because at the bottom of the tube, you have your most concentrated fluid. It's still pumping the sodium. It's got all this extra sodium to pump out. That's why you have the gradient going down. It gets more concentrated as it goes down. You hit the bottom of the loop of Henle, it's the most concentrated gradient, about 1,400 milliosmoles.
Is that basically the countercurrent multiplier then?
Yeah.
It's that one side.
Well, the countercurrent is when one's pumped out, it's drawing, but as it's pumped out, it becomes more concentrated, so it's easier to pump out. Then, so you pump out more, so it becomes more concentrated, and it just builds on itself until you hit the bottom of the loop again.
Okay,
Okay?
got it.
Because it's easier to pump it out because it's more concentrated. Okay? And it's more concentrated if it can flow through the proximal convoluted tubule, then it's still going to be drawn out too. And out it goes. Okay, loop of Henle, solute and water transport. Descending, you've got water going out, you've got sodium that's being pumped out, you have descending passively permeable to water, ascending, active transfer to sodium, chloride follows, it's impermeable to water. So you've got this concentration here, gets the most down here, you've drawn all your water out, you go back up. If this was permeable to water, water would be flowing back in, but it's impermeable. You're pumping out the sodium, so it still becomes more dilute as it goes up. Not because it's being diluted out by water, but because the sodium is being pumped out.
The urine.
The urine. The urine, yeah. Countercurrent multiplier, positive feedback mechanisms. More salt, the ascending limb removes the saltier the fluid. Entering it will be due to the loss of water in the descending limb. The more salt that comes out of the ascending limb, the more water is coming out of the descending limb. That's your countercurrent. Vasa recta, specialized blood vessels surrounding the loop of Henle, have a descending and ascending. They help create the countercurrent because they take in the salts in the descending region but lose them in the ascending. Keep the salts in the interstitial space. If they take the salts in, the water is going to follow it in. High salt concentration at the beginning of the ascending pulls water, which removes it from the interstitial space. The water comes out of the nephrons into the interstitial space. The vasa recta has taken the salt and it's pulling the water in. You don't want the water in the interstitial space. You want it back into the capillaries. So this is the same thing you saw earlier. Urea, waste product protein. We are not going to get into that. I don't think he's going to get into that. Renal tubular transport properties. Renal tubular osmolality. We just showed you that already. Collecting duct and ADH. Last stop in urine formation. Influenced by the hypertonicity of the interstitial space, water will leave us if able. Permeability to water in the collecting duct changes the number of aquaporins. What ADH does is stick aquaporins into your collecting duct to allow water back in.
Supporters.
And if you're real hydrated, it's not going to let water back, it's going to let water back in. If you're dehydrated, it's not going to let water back in because it wants the water to go into the veins and back into the circulation. Anti-diuretic. Anti-urine formation. Okay? A diuretic causes you to pee more. Anti-diuretic keeps you from peeing. That's because the number of aquaporins it allows into the collecting ducts.
So the more aquaporins, the more you don't pee.
No, the more aquaporins, the more water is going back into the collecting duct and you pee more. Okay? So low ADH, you've got more aquaporins. High ADH, you've got less aquaporins.
So when you drink, it makes your ADH go up, so less aquaporins, and you pee more,
Yeah.
right? And you get dehydrated.
And caffeine and alcohol affect that too. And so that's why you pee.
Caffeine works the same way?
We may get into it. It's something like that though.
I feel like when I drink, I use more.
You're supposed to.
Okay. You get dehydrated. Okay.
Because you're well hydrated and your body isn't trying to conserve the water. So your ADH is not diuretic, it's anti-diuretic. So if it's up there, you're not putting the aquaporins in. So if you're drinking a lot, your ADH is low. If you're not drinking and you're dehydrated, your ADH is high, and that takes the aquaporins out so that you're not urinating off the water. It's going back into the system.
And that's all regulated by your endocrine pancreas releasing the ADH.
Well, it's regulated by the baroreceptors, but the ADH is released by the anterior posterior pituitary.
Okay, so that has to do with, I don't want to get in because but it has to do with calcium and potassium stuff too, right?
It's all in the, this is the big picture. You can get into the minutia, but I don't think he is, and we could spend all day on this chapter. Okay? Are we going on? Collecting duct. ADH inhibits receptors. Okay, collecting duct, here's just putting your aquaporins in. Role of ADH in plasma concentration, it's in your book. You've got your osmoreceptors, you've got your posterior pituitary affecting the kidneys on water reabsorption. Okay?
It's page 590 of this book.
Summary. ADH, this is 17.3 in your book. But you need to know ADH. It's on here. Filtration, reabsorption, diuretics, kidney disorders. Renal plasma clearance, we are not going into here. Secretion, you have secretion. Oh, actually, we are going to go in here. Excretion, filtration, we're not going into that. Secretion, remove excess ions and waste from the blood. Filtration, the glomerular capsule begins. Secretion finishes when the substances are removed into the tubules. Oh, wrong way, sorry. Excretion. Reabsorption of glucose, easily filtered out, completely reabsorbed in the proximal tubule. That's a great test question. Where is glucose reabsorbed? It's reabsorbed in the proximal tubule. Secondary active transport, sodium facilitated diffusion and simple diffusion. As it pulls sodium and that in, it pulls the glucose in with it. It's secondary, remember what secondary transport is. It pulls one molecule in, another molecule is attached to it.
Like NaCl?
Yeah.
Wait, reabsorption of what? Glucose?
Glucose and amino acids, they take place in the proximal tubule.
Through secondary transport?
Yes. Transport saturation, at some point you can't. Oh, I'm sorry. We didn't cover that for you?
Hi, Dylan.
Secondary transport. At some point, you can't absorb all the glucose. Usually that's above about 140 milligrams per deciliter. And it goes through it, and after that it's going through the descending and ascending loop of Henle and out. That's glycosuria. If the blood sugar is so high that it can't reabsorb it, if so much glucose is going into the filtrate, can't reabsorb it, it's going out in the urine. That's what you see in the diabetics.
I thought that you guys were diabetic.
Well, that's one of them. Extra glucose in the blood also results in decreased water reabsorption and possible dehydration, because as the glucose goes out, it also creates in the inside of the descending proximal tubule, it doesn't allow that complete osmotic gradient, because glucose provides osmotic pressure in the tubule. So not as much water will go out to be reabsorbed.
So it should stay in the water, yes.
Now, what is typical of a diabetic? You get up in the middle of the night, you're drinking and peeing all night long. That's because the glucose doesn't allow the water to go out of the proximal, or the descending loop of Henle.
So if you're peeing all night long, could be a sign that you have diabetes?
Absolutely.
I never knew that.
Just figure out.
Yeah. That is one of the classic thirst. Thirst is, before you can check your blood sugar, if you are drinking constantly and constantly thirsty, you're going to get checked for diabetes. A friend of mine, daughter came down with juvenile diabetes, and he got an idea when he would walk into the bathroom in the morning, and there would be 7 or 8 paper cups that she'd been in to drink water all night long. And they thought, they brought her in, her glucose was about 600 when they brought her in.
Oh my God.
The same with animals. If they're constantly drinking all the time.
Could be.
Could be.
My dog never drinks.
Well, you may die of...
Maybe I'm just not.
Dogs don't sweat either. So.
Okay, let's go on.
My dog does.
But you got that?
Sweat.
Why? Great question. Why would a diabetic not reabsorb water? Why would a diabetic pee as much?
Not.
Pee more. Why would they
Because
pee
the
more?
osmotic pressure inside the tubule because the glucose has gotten through.
Exactly. The glucose gets into the descending loop of Henle, and it creates, it does not allow, the salt is
The
up.
water
The
can be
glucose stays in there. So it creates an osmotic pressure that's going to try to draw water back in or let less water go out, which means that water stays in the loop of Henle and gets peed out.
Yeah.
Okay? Let's go on. Kidneys match electrolyte bicarbonate excretion to ingestion. Okay? You take in a lot of salt, you're going to pee out a lot of salt. That's what happened in our labs. Remember the chloride?
I don't think so. I think it's you can go.
Aldosterone.
No, he doesn't. He said it's not going to be on the test.
Really?
Yeah, he said aldosterone, he doesn't.
Go back to that.
Just ADH. ADH is the only thing that's going to be on the test.
Kidneys match the electrolyte concentration of the excretion to ingestion. Your electrolyte, when we measure it, are both in very narrow ranges. Your kidney is the one that controls that.
Because of the ADH, right? Or
Not necessarily.
isn't that for...
Actually, it's more aldosterone than it is ADH. ADH controls the fluid.
I mean, yeah,
But we're not going into aldosterone, so I'm skipping it. We don't have time to do it.
You don't think he'll ask us about blood pressures and stuff?
corticosteroid.
Just know that the kidney.
The kidney regulates it by regulating the fluid volume in the body. I don't know what else he's going to ask.
That's what Sarah said in SI, based upon other exams that she did.
Potassium secretion, aldosterone, aldosterone independent, aldosterone, potassium, increase sodium absorption, decrease. This is a take home. The more sodium you absorb back in, the more potassium is going out. It goes vice versa. If you are very hyponatremic, you have low sodium, your kidney is going to absorb as much sodium, but you are going to bleed potassium. You are going to pee potassium. Believe me, you're going to lose it. Not necessarily, and I know it's a wrong class. It is a wrong class to say something like that.
So you're going to lose...
You're going to lose electrolyte. If you are very hypernatremic, your sodium is very low for whatever reason, your body is going, your kidneys are going to reabsorb sodium, but they are going to do it at the expense of potassium.
Because it's a positive ion?
It's just an exchange. Okay?
My mother-in-law doesn't absorb potassium.
What's that?
She does get potassium. Like she has to. She has to increase in her diet, but
Is she on diuretics?
It just a couple of years ago she started fainting, passing out, and she went in. Her potassium levels were almost non-existent. They said, "You're going to die."
You are gonna die.
Yeah.
You are absolutely gonna die. Potassium is a very narrow
She
one.
tries to get it through her diet more than anything.
Bananas?
Yeah, so she doesn't have to.
But,
Oranges too.
well, the reason it's oranges is because when glucose goes into a cell, it pulls potassium in with it.
Right.
And what happens is it may not raise your blood potassium so much, but it does raise the overall body potassium. So you store a lot more potassium. If I get somebody coming in and their potassium is at 7 or 8, which will kill you very quickly, what we do, I mean, there's now some potassium reducing drugs, but the first thing we're going to do is give them a big bolus of glucose and a big bolus of insulin. That's going to suck all that glucose into the cells, and it's going to take potassium with it. It will lower that serum or blood potassium very quickly.
So,
What is their blood glucose go to though?
We don't worry about that because that's not going to kill you acutely.
That's true.
The potassium will kill you in a couple of minutes.
Right.
so is potassium, since it's happening throughout the loop of Henle, potassium is controlling what's happening, or is just allowing things to go in and out?
No, it's not. All I'm saying is potassium isn't controlling. It's the aldosterone, it's the whole body balance. But if you are sucking in, if you're reabsorbing sodium, you're going to lose potassium because they're exchanged.
Okay, that's it.
Okay? Let's go on. I don't think we can have that on here. It's not time to tell stories. So,
That makes sense though. I mean,
control of aldosterone, no. Juxtamedullary, angiotensin,
no,
That's it.
renin, no. Macula densa,
no,
no.
no.
Atrial net, sodium, plasma sodium balance, we've already talked about that. Regulation of aldosterone, no. Relationship between sodium and potassium, no. Well, no? No.
That's all.
Acid-base regulation, we got it, right?
We got it.
We have been over this many times.
Bicarbonate.
Bicarbonate doesn't cross. It's the hydrogen that's excreted or reabsorbed. And actually, bicarbonate is broken down and then it's reformed, but we're not going to get into that.
Nope.
See, you can't reabsorb it. It's got to be broken down and then it's reformed. Secretion of hydrogen, sodium hydrogen pumps in the proximal tube. pH disturbances, alkalosis, we've talked about. Less hydrogen available to transport bicarbonate in the tubule. Bicarbonate is reabsorbed. pH disturbances, acidosis.
No.
pH, urinary buffers.
No, we're done.
Clinical application. Diuretics is on here.
Oh, you know what?
Clinically used, what's that?
You guys talk about,
Diuretics, clinically used to control blood pressure. Some by pushing out more sodium, excreting more sodium. And diuretics increase urine volume, decreasing blood volume, interstitial fluid volume. They affect different portions of the nephrons.
so antidiuretic decreases urine volume, right?
Yes, antidiuretic decreases urine volume. Loop diuretics, most powerful, inhibit salt transport out of the ascending loop of Henle. What does that do?
That makes the great osmosis inside the loop, so water stays.
Yeah, you don't have as much gradient for the descending loop of Henle to draw water out. So you're going to pee more. That is exactly kind of the same thing that glucose does, except this just keeps the salt in. Okay?
So, I don't know if this is way off, so now potassium would be, would that?
Forget about potassium. Okay? Right now, just forget about potassium. You will take us in the wrong direction.
Can you flip
It's
it
hard
back
to
the
though,
other
because it's
slide?
actually really important, obviously.
Is it important for the test?
No.
Can you flip it back to the other
Important
slide?
if you're going into nursing stuff.
The other
We want to go back
diuretics.
one?
Yes.
Diuretics increase urine volume, decreasing blood volume, and interstitial fluid volume. If the interstitial fluid volume goes up, it's going back into the blood. Diuretics keep the water from going out into the interstitial fluid. Loop diuretics, Lasix.
So,
We're done with this chapter.
What?
We're done with this chapter.
You didn't go into it beyond that?
No, we're done.
You have two different, loop and thiazide.
no. Where is it?
Acute renal failure?
No.
Actually, that is
A little
in the
bit.
slides,
Kidney disorders.
Renal failure, glomerulonephritis, and renal insufficiency.
Glomerulonephritis, inflammation. It's autoimmune, or it's from streptococci, antibodies to the streptococci.
Yeah.
You have strep throat, there's two places the body's going to make antibodies and it's going to attack. It's going to attack the nephrons, and it's going to attack the heart valves. That's your rheumatic fever, and you get a glomerulonephritis. That's your most common, in kids, reason for this disease, and they can lose their kidneys. Their kidney function can go way down. That's why they treat strep throats so aggressively. Let's go on. Renal insufficiency is reduction in renal capacity. High blood pressure, high potassium, uremia. This is where you do dialysis. Okay? Done with the kidney. Questions?
Moving on.
No, we hate the kidney.
Thank you.
Okay.
What, Steph? Did you have one? Me too.
We're just so done with the kidney.
Actually, the kidney's kind of...
Nephrons are awesome. They are, but we're so done with going over them and that's
So
all.
you're definitely not going to work in neurology.
I'm sorry, but we're so nephron.
Okay, let's go on.
You'll end up loving it when you start.
Digestive system.
Digestive system.
Poop.
Oh,
Bolus poop.
Okay, I took all these slides, excuse me, the ones that I knew we weren't going to do, I tried to just delete, but then after a while, it just put them in, we'll just pass them by, because it was getting late. Okay, open at both ends. Your digestive system is extra body, it's outside the body. Okay? Materials that cannot be digested, they never actually enter the body. They are pooped out. Digestive tract functions. You have motility, ingestion in the mouth, mastication, chewing. Degglutination is your swallowing. Peristalsis, one-way movement through the tract. Down the esophagus, through the stomach, through the small intestine, through the large intestine, and out. Segmentation. Churning and mixing, that is part of your peristalsis. Some of your peristalsis moves it through, some of it just mixes it. Okay? That's your segmentation. Segmentation is the mixing in the intestines and in the stomach. Secretion. Know your endocrine, exocrine, paracrine.
Oh, that's right.
You got to know that because it comes up through all of this stuff. Endocrine goes where?
Inside.
Into the blood.
In the blood.
Exocrine goes
Outside.
into a space.
Yes.
Paracrine in itself and cells right next to it.
Okay.
Okay? Those are your hormones. Exocrine, digestive enzymes, acid, mucus. That's in your stomach.
Inside.
Your stomach's a nasty environment. Endocrine, hormones to regulate digestion. Breaking down of digestion is breaking the foods down into smaller units or monomers. Absorption, passing the broken down fluid into the blood or the lymph. Storage and elimination, it's temporarily stored in your large intestine and eliminated after that. Immune barrier. Simple columnar epithelial, they have tight junctions for really prevent a lot of the swallowed antigens, swallowed pathogens. In your mucosa, in your submucosa, in the intestine, you have what they call
Laura. Wait, what's the question?
Peyer's patches.
What is that for? Bacteria?
That is immune responsive tissue. So it can recognize foreign particles and make antibodies, or cause the body to make antibodies. Okay, it's protective against pathogens coming in through the gut.
Is this tissue?
So detects pathogens.
It's active immunologically active tissue in the submucosa, I believe, of the intestine.
The ileum. It's also where the valve opens to protect it from the other stuff going back up, right?
No, I don't. Byers patches it's part of the absorption. It's not in the intestine.
They're actually in the
It's
very
under the
end.
layer.
It's at the very last end of the section of the ileum before the ileocecal valve, where
Okay.
it goes from small intestine to large
The
intestine.
large, yeah.
So the
But the
bacteria.
valve that's not part of the valve isn't part of the pyre patch.
No, but the poop that's going into that's in the large intestine, so it doesn't, the bacteria from that doesn't come back up through.
Well, that's not part of that's part of the valve, not Peyer's patch. The Peyer's patch is for an immune response.
Right, so that bacteria that could enter through the valve going backwards rather than forward, that's.
I think that would be a stretch.
So.
See, I thought that too. I thought it was the stuff coming from the gut before it enters the large intestine, but then I was told it was the other way. Move on.
You may want to look. I honestly can't answer that for you.
So the pyre patches are like indicator for the lymph
That's
system?
Not indicating. The lymph system is an immune active area. So are the pyre patches.
Oh, okay.
Okay?
Okay, so they're.
They're immunologically active, meaning they can recognize foreign and cause antibodies to be made. Okay? Beyond that, where the foreign stuff's coming from, I can't tell you.
We're going to move on.
Okay.
Is it only in the small intestine? Or is it?
I think it is.
The
I don't
Peyer's
know
patch?
for sure. The Peyer's patch.
You said just a small intestine.
Yeah.
Can they attack directly?
What?
Can they attack directly?
No.
Oh, sorry.
No. Let's go on. Digestive divisions: mouth, pharynx, esophagus, stomach. If you're in a cow, you've got 4 of them. This was 2 slides that kind of went together.
I kind of like that. Can I read that?
Well, if you want to say you've got 2 stomachs, okay. Small intestine, large intestine. Okay, accessory organs. I think you wanted to know what some of those were. Teeth, tongue, salivary glands, liver, gallbladder, pancreas, hormones, mastication, swallowing, all that. GI tract, tunics, 4 tunics. Mucosa is the inner submucosa muscularis and serosa zeae.
I have a question. That is on all the GI tract, huh? From the starting of your esophagus all the way to your anus, huh? I know it's in the stomach. All those feelers are in the stomach.
Yeah, I think it probably does go
Yeah, all
over.
through the GI tract, I noticed that.
Regulation, parasympathetic division, stimulates esophagus, stomach, and small intestine. Parasympathetic, you really have no control over it. Stimulates esophagus, pancreas, gallbladder, first part of the large intestine via vagus nerve.
Are these called visceral organs?
Visceral?
Visceral organs, or?
Yeah.
Yes.
Yes, this is all part of your visceral.
Organs. That's why it's.
Visceral, yes. Spinal nerves in the sacral region stimulate the lower large intestine. What would be happening with that? Yeah, peristalsis, the excretion. No, the only thing that's absorbed, that happens is storage and absorption of water in the lower intestine. In the large intestine, excuse me. Preganglionic neurons, synapse on the submucosal, and mentoric plexus. Keep going. Sympathetic division inhibits peristalsis and secretion, stimulates the contraction of the sphincters. Some of the stuff you can control to a certain extent. Your sphincters are one of them, to a certain
So
extent.
it's like an internal and external, and that's what little kids have to
Learn to use.
learn to, yeah.
To know the difference?
Yeah.
Is that why my daughter pulls her food? Okay, so sympathetic inhibits peristalsis. Okay.
Yeah.
You guys
Okay.
want to hold it, or?
Well,
Inhibits.
okay, going on. Intrinsic regulation, sensory neurons in the gall. Well, gut wall help. As the gut expands, there's neurons that sense it. As the stomach expands, as the chyme, you know what chyme is? As chyme moves down from the stomach into the duodenum, it expands, there's sensory neurons, it excretes bile, urine, the blood insulin is going up in expectation of the sugar that's coming in. Okay? That's part of your paracrine signals. Stomach function, stores food, churns. Is that segmentation? Segmented peristalsis? Churns. Mixes with gastric secretions, begins protein digestion. What's in there? Pepsin. Pepsinogen is excreted in the lower pH in the stomach, turns it into pepsin, which breaks down your proteins. Kills bacteria, not all of them, but some of them. Moves food into the small intestine in the form of chyme.
Remember those illustrations that showed how the stomach, and there's several things that cause everything. There's like three different things, three different positive feedback loops. It's how the brain, and the brain's another thing down there, and it starts shutting off the.
Well, there's three different areas of the stomach that do different things. But let's go on. Stomach structure. The upper division is the fundus. Lower body distinct is the pyloric. It ends at the pyloric sphincter. If you have had a rule in Y, they have removed the pyloric sphincter. Makes it hard to control. That's all part of the gastric bypass. Lining has folds called rugae. The more folds, the more surface area for the excretion of enzymes. In the stomach, there's only a couple of things that are reabsorbed. Alcohol is one.
And drugs.
And what?
Incense.
Drugs and alcohol.
Incense?
Incense. They are called
Motrin, ibuprofen.
No, well, that too, but that's a whole different problem that we might get to.
Well, what were you thinking?
Yeah.
There's another organic thing.
We're thinking beer?
Well, that's the alcohol. Alcohol is absorbed right through the stomach wall.
Organic,
The
huh?
house.
We'll probably get there. There's a part you need the stomach
Oh,
structure.
that looks gross.
No.
Gastric pits, they lead to gastric glands with secretory cells. The idea of the secretory cells and your gastric glands is your acids have to be activated after it gets into the stomach. If it's activated before, it's going to eat your stomach wall up. And then you've got a what?
Ulcer.
Ulcer. The parietal cells secrete HCl, intrinsic factor. We talked about intrinsic factor. Vitamin B12 absorption. You don't have intrinsic factor, you don't have vitamin B12. You don't have vitamin B12, you have pernicious anemia. Pernicious anemia, we treat with vitamin B12 today, in the 20s and 30s. Before that, it was a Kisadel.
Can you not change the slide yet?
I didn't realize parietal cells did that.
Yep.
He just wants you to know HCl and parietal cells, that's it.
Okay.
And chief cells and
Chief cells secrete pepsinogen. The parietal cells secrete hydrogen. The hydrogen breaks down the pepsinogen into pepsin, which breaks down protein. That's how it works.
Okay, say it again. Pepsin does what to pepsin? I'm sorry.
Hydrogen.
From the HCl.
Hydrochloride. Low pH.
Okay.
Breaks down pepsinogen into pepsin.
Okay.
Okay? Pepsin breaks down proteins.
Okay.
Starts the digestion of the proteins.
The more H, the more acid.
And, well, the more hydrogen, the more acid, but the more protein, the more buffer. So it doesn't drop down, so you get more acid, so you get more pepsinogen. That's how the stomach regulates on how much protein is coming down. It's, again, almost a convoluted theory.
It breaks down so it's not- it breaks down so
If you have more protein in the stomach, it buffers the hydrogen. So your body's going to have to secrete more hydrogen to get more pepsinogen to change the pepsin so you can break down the protein.
So it breaks it down so it's easy.
We're getting off track.
Oh, okay, easy.
We're getting off track. Pepsin, pepsinogen to pepsin. Pepsinogen plus hydrogen goes to pepsin. Pepsin breaks down protein. Okay? That's the take-home message. In the stomach, you need the low pH. ECL cells secrete histamine and serotonin.
No.
G cells?
No.
D cells?
No.
On. Stomach structure? On. HCl secretion.
No, he says that's not.
Maturation?
That's
How
not
the
what
hell did
you-
that get in there? Okay, stimulation of HCl. Gastrin, G cells? No?
No.
What does he have?
Each one says no, G cells are fine.
Regulation, digestion, stomach, small and large intestine. Okay, we're going on. Function of HCl, we already talked about that. Stomach defenses, you've got the mucus layer, you've got bicarbonate, you've got epithelial cells that are replaced about every 3 days. The stomach lining replaces itself. Proteins begin digestion in the stomach, starches in the mouth. Saliva, amylase. Start your digestion of starches in the mouth. The amylase, other than that, comes in the intestine. Alcohol and aspirin are the only common substance absorbed in the stomach.
Oh, see, they said NSAIDs was aspirin, that's what you were thinking, huh?
Something like
I
that.
thought it was Motrin.
Peptic ulcers, erosions in the mucosa. 80% of your ulcers are from Helicobacter. Now, the other thing about NSAIDs is it decreases the prostaglandin that the stomach needs to replace the mucosa. That's why aspirin, not Tylenol so much, but aspirin and ibuprofen are terrible for the stomach. But even though it's absorbed through the stomach wall, it doesn't activate until it gets into the blood and inhibits the prostaglandins. You can give an aspirin suppository and have stomach problems.
Wow.
Wow.
Chirpy.
Yeah, it is. Kind of working from the wrong end. Okay, treatment for ulcers, sodium, you have Prilosec, you have proton pump inhibitors. They inhibit the amount of gastric, the amount of hydrogen that goes into the stomach. That's your proton pump inhibitors. Gastritis,
Can we go back to that proteins aren't being broken down?
it means that not as much acid is getting in. The acid is what's eating away at
Still
the...
getting some acid to break.
You're still getting some.
Okay. Can you go back just for a second?
That's how far back I
No,
can get.
no, just that one slide. Yeah, wait, back.
What?
The one right before that, you were just on it.
Right there?
The pump one, the K and the,
Yes.
thank you, treatment for ulcers, okay.
Prilosec and antibiotics. Antibiotics kill the H. pylori. Prilosec decreases the amount of hydrogen that's pumped into the stomach. Proton pump inhibitors. Okay?
Yeah.
Well, Prilosec or Peps. Is it Pepsin?
Or commodity,
I don't know.
well, same thing.
Inflammation of the submucosa caused by acid histamine release. Prostaglandins are needed to stimulate the alkaline mucus.
I think you can move on.
That's what your NSAIDs attack.
Oh, NSAIDs.
Small intestine. Here we go. Three sections. Duodenum, jejunum, ileum. Mucosa, submucosa are folded into villi and microvilli. Why? More surface area. More surface area for reabsorption.
All he wants to know for small, large intestine is what food molecules are absorbed.
Here we go. Okay, small intestine, digestion of the fats, carbohydrates, and proteins. Absorption, sugars, lipids. You guys want to take a picture of this? Sugars, lipids, amino acids, calcium, iron, absorbed in duodenum and jejunum. Bile, vitamin B12. Water and electrolytes in the ileum. Very rapid due to the villi and microvilli. Huge amount of surface area. Your wrist getting sore? Should I put this on YouTube tonight?
You probably should, my gosh, because I'm not.
And for all the people that didn't bother to show up, I'm going to eliminate that comment before I put it on. Probably not.
You just copy it to my scanning.
I know.
No, I'm not doing that this time.
What did you do last time?
I gave you a whole bunch of answers to questions. Maybe we're not on the desk, however.
Oh, my goodness.
So no, that ain't happening this time. Villi and microvilli. Come, you got that? Absorb
More
sugars,
surface
amino acids, all
Surface
that.
area, got it.
Villi and intestinal enzymes. Brush border enzymes. That's a good question. What's a brush border enzyme? It's an intestinal enzyme. Not released into the lumen. They stay attached to the villi walls. They work on the walls. They don't go in and into the lumen and cause a chemical reaction. The chemical reaction takes place right on the wall of the villi. That's a brush border. You ever heard about brush border wars? This, yeah, like Africa, they always say we all get the brush wars and brush border wars. They're right on the border, right on, attached right to the end, right to the villi. Okay,
They
Keep
are.
going.
Keep going.
that's in your book. You want to watch it or not?
No.
Okay. In contractions, motility, peristalsis, segmentation. We've already talked about this. Peristalsis moves it, segmentation mixes. Okay? Intestinal contractions, there it is right there. Contractions, smooth muscle contractions automatically. Graded, you have graded depolarizations. Interstitial cells of K-Hall. Got that?
Yeah.
Okay.
No, go.
Contractions, contractions, contractions.
Contractions.
Large intestine.
Okay.
Chyme from ileum passes through the ileocecal valve. They never could get all the spaces in there. Okay? Here it is. Not a big deal, but it absorbs water and storage. Absorption of water, electrolytes, vitamin K and some B vitamins, well, mostly water.
Water and what else? Storage?
It'll absorb some water and electrolytes. It's not absorbing nutrients. K and B vitamins. Storage of yeast.
Bless you.
Thank you.
It produces K and B vitamins because of the work of the E. coli and some of the bacteria in them.
Oh.
Microbial. Large bacteria, commensal, might benefit from microbes, K and B vitamins. Absorption of fluids, large intestine.
Aldosterone.
Wrong.
Is that
Go. Keep
A lot
going.
of
Defecation.
It did say
just about
aldosterone. Keep
sphincters
going.
relaxing?
Okay, sphincters relaxes.
What does it say about aldosterone?
Internal.
Nothing in there. Liver, gallbladder, and pancreas. What did he want on that?
Those are our main.
Accessory organs.
They're accessory organs.
The function of each one.
Liver stores bile and does another thing. One important thing. Sarah does.
Detoxifies.
Detoxifies many people.
It detoxifies, it stores bile, it stores glucogen, it makes cholesterol,
It stores
Let's-
glucose in the form of glucogen.
glucogen,
Glycogen.
glycogen,
Stores glycogen.
Glycogen.
and glucogen.
Don't tell me that.
Hang on, let's go. Detox, carbohydrate, converts blood glucose to glycogen and fat. Okay? Production of glucose from liver, glycogen, and other molecules secretes glucose into the blood.
What's the last part of bile? What's it?
Of bile,
Secretion of bile?
conjugation and excretion of bile pigment, synthesis of bile salts. What are bile salts made into? What's the big thing that comes out of bile salts? Cholesterol. But it did start with, there had to do with the C or something. Okay? I'm not sure we would give it to you, but
Triglyceride.
good luck. That's not a
Triglyceride?
triglyceride, that's fat. Okay, cholesterol is the building block of your hormones and things like that.
That's structure of your
Structure is what?
It adds structure.
Yeah, it adds structure. Okay, so carbohydrate metabolism, lipid metabolism, protein synthesis. This is where you get your albumin. What does your albumin do? What's the main purpose of albumin?
It's
No,
for eggs.
it's part of albumin, it's in your blood, isn't it?
It's in your blood in a very high concentration. It creates the osmotic gradient to draw fluid back into the blood and the lymphatic system.
Oh, it's like the egg, right? The egg. The egg.
That's
It's
what I
kind of like the egg again.
Oh, yeah, but it's
We want all the water out of the egg.
Well, when we put it in distilled water, we threw it back in, the albumin in the egg didn't.
Oh, yeah, it's
But he may accept that on the test. Protein synthesis, production of clotting factors. Beet bile production makes 250 to 1,500 mils a day. It's recirculated, it's reabsorbed. Bilirubin, breakdown from red blood cells. Carried by albumin, not filtered in the kidney or secreted. Well, conjugated, it makes it water soluble. Conjugated is secreted. Did he go? Does he want to go?
No.
No.
Don't even bother. Don't even bother.
secretion of glucose.
Removes hormones, drugs, and other substances secreted into the bile, phagocytized by Kupffer cells, chemically altered. Okay? Ammonia is converted into urea. If your liver dies, your ammonia goes through the ceiling. It's one of the things that we watch very carefully.
What? Urea.
In liver failure, your ammonia goes through the ceiling. Secretion of glucose. Glucose is stored in the liver as glycogen. What's the difference between the glycogen stored in the liver and the glycogen stored in the muscles?
Because that's for a reserve when there is no nothing.
They're both for reserve.
Well, the glycogen in the muscles does not come out of the muscles.
Right. The glycogen in the muscles does not come out of the muscles. It can only be used by that muscle. The glycogen in the liver from glycolysis can be excreted into the blood to provide blood sugar for all parts of the body. In your fight or flight syndrome, that goes right through the roof. Your glucose goes up because of the need.
Via the blood.
Via the blood, via the liver and the blood. Gallbladder stores bile from the liver. What does the bile do? Emulsifies fats. You need anything more than that? You need the...
No, just skip it.
Can you go back to 202 back? Because there was something about, yeah,
Don't need it. Keep moving.
fatty acids.
Don't worry about it.
If you have a lot, you're eating a lot of protein and not a lot of fat or carbohydrate, you can still make fat. You can still make glucose out of gluconeogenesis. Gallbladder, pancreas. What's the main thing the pancreas does?
Insulin and glucose.
Insulin, alpha and beta cells. Okay?
Beta is insulin.
Yes.
Beta cells.
Alpha is the
Glucagon.
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 Bicarb.
Just think of insulin as baseline.
That's a good one, Ro.
Bicarb, made by cells lining the ductus, made CO2 from the blood. Obviously, it's first carbonic acid. We've been through this many times. Bicarbonate is secreted in the pancreatic juice. Hydrogen goes back into the blood. Why? Just completing the circle. Bicarbonate is secreted into the pancreatic juice, which is secreted into the duodenum, right up by the pyloric sphincter. Why?
So it's not so.
So it unacidifies the chyme.
It picks up the hydrogen.
It neutralizes. The chyme coming out of the stomach is very acidic. The duodenum cannot handle the acid. So by the time the bicarbonate is pushed in, your chyme is essentially neutral in the duodenum. Pancreatic enzymes, trypsin, trypsinogen, trypsin, enterokinase.
There you go.
Neural and endocrine. Neural control modifies GI tract functioning. You have smell, stimulates it. Brain via vagus nerve. That's the main nerve for that. Motility and secretion are somewhat automatic.
Secretion of AUC.
When amino acids enter, when the protein gets in, you're going to secrete HCl and the pepsinogen. Okay?
Intestinal.
But you're not going into the G cells, D cells, and ECL cells, right?
No, keep moving.
Skip one.
Extrinsic.
Skip that.
Extrinsic.
He skips, yeah, there you go.
Controlled by the brain via the vagus nerve.
That's 2G. Cephalic, see, smell, taste, and then gastric is the
Secrete,
stomach.
secrete, secrete.
The secrete of stomach juice, right?
From the G cells, gastrin.
Yeah. And then-
HCl and pepsinogen when they sense the amino acid. Gastric phase triggered by the arrival of food, divided into 3 phases.
Too deep.
Gastric secretion by food, negative feedback as pH drops due to more HCl, somatostatin is released, inhibits gastric secretion. Lots of proteins, as I said, buffer the pH so the secretion matches the protein concentration. 3 intestinal phase, inhibition of gastric activity when chyme enters the small intestine. What inhibits it? Bicarbonate. Stretch. When fluid enters the duodenum, there's a neural stretch via the nerve for the excretion of the bile for all the other enzymes. And then fats enter the enterogastro. Yeah, no.
Skip. Thank you.
Intestinal function, enteric nervous system. Are you going to go into this?
No, just know that the sympathetic is, or the parasympathetic is rest and digest, right?
Yep.
Okay, skip.
Parasympathetic is rest
So
of the
it's
nuts.
for the
Yep.
intestinal disorder.
Sympathetic is fight and flight.
Yeah.
What she talked about?
Regulation of intestinal function. I think we've been through that. Paracrine.
Skip. Yep. Skip all that. Yep.
Regulation of metabolism.
That was pretty good.
We got through that chapter in 30 minutes.
Yay.
Okay, we'll go for about another 25 minutes and then take a break because we're going to have to move to another room. And we'll move down to 106 at the end.
Why do we have to move these?
There's a final in here.
No. That's not right.
No.
And I would bet if we continued, it would disturb them.
Yeah.
It's possible.
Okay. Regulation of metabolism.
Okay.
Nutrients, nutritional regulation, calories. The big thing you want to know, what's the difference between a small C calorie and a big C calorie?
1,000.
1,000. It's a kilocalorie. You want, if I put this on YouTube, you're just not going to have time to look at it again. Get your husband to cook and clean for a couple of days while you're studying.
Word.
Okay, energy from ATP. Calories. 1 kilocalorie is 1,000 calories. When you look at calories on a calorie label, they're generally talking about kilocalories. Calorie raises water. Good luck. Have a good one. Calorie raises water, 1 mil of water, 1 degree centigrade. It's enough energy to raise 1 mil of water, 1 degree centigrade.
Celsius, right?
Centigrade and Celsius are the same.
Okay. I need to know that.
Okay. Metabolic rate measured by the amount of heat generated. You got to tell me what he wants in this. 'Cause I don't have a
I
clue.
don't think those numbers.
Major nutritional.
Regulating metabolism, adipose tissue for fat,
Yeah.
liver tissue for sugar. We've already touched on that. Okay, let's just go through. Anabolic, food required, raw materials. The stuff that you cannot make are essential nutrients. Essential nutrients, lysine, tryptophan, phenylalanine.
Make it
You
go
gonna know that?
Actually, that's one of the questions is what are our major nutritional requirements?
Yep.
It would be essential things.
Yep.
Essential means we can't make it, we have to get it from our diet.
Right. Small organic molecules or vitamins. They assist in various chemical activities. They can be like a subunit on an enzyme. That may be some of your vitamins. fat soluble and water soluble. Minerals, large amounts, sodium, potassium, magnesium, needed small amounts, zinc, manganese, fluorine, selenium.
Too deep.
Keep going.
Free radicals.
Stop. There we go.
Did he talk about free radicals?
Yes, he did.
Free
Okay,
radicals are very important.
very important. Why? They destabilize synthesis. A free radical,
Just get a crazy guy running in your body.
well, you know, the free radicals, when you're trying to synthesize proteins and other, may knock off a gene or something here and there and cause mutations. They knock off the wrong ones, they knock off the one that inhibits cell growth, and all of a sudden you've got cancer. So free radicals are not a good thing. That's why they're always telling you to eat blueberries and things like that. Because it adds an antioxidant that takes care of these. Very reactive in the body, oxidant or reducing other molecules. You got it? So a role in immune response in phagocytic cells, vasodilation, scar formation.
Okay, so does that mean we need free radicals as well?
Too many cause oxidative stress on cells, damaging proteins, DNA and lipids.
But we
You
need
do
them no
need
matter
some,
what.
but you don't need a lot. Okay?
Just stop what everything should be doing, just supposed to do.
Well, they have things formed wrong.
Yeah.
You know, if it gets into the DNA, you realize you start knocking off various sequences out of the DNA, and if something that doesn't pick up, then you're going to end up with a mutation or something that doesn't work,
Radicals are just like loose electrons, right?
essentially. Antioxidants, enzymes, catalase. Is he going into this, or do we need to spend a lot of?
Just know the antioxidants.
You need to know what
The
antioxidants
free
are.
radicals.
Yeah.
Okay, vitamin C and E, pick up unpaired electrons. Other molecules in fruits and vegetables are antioxidants. That's why they tell you to buy the blueberry, huge in antioxidant. Pomegranate, blueberry,
Coffee.
no,
Dark veggies and
dark chocolate, dark chocolate.
Always look for dark.
Free radicals, this is in your book, I believe.
Too deep. Keep going.
Regulation of energy, this is where glycogen and fat are good, proteins may be used in emergencies
So really for energy, it's just glycogen and fat?
until you're starving. Then it'll break down protein.
And
Protein's the last thing that's going to be broken down in energy.
So glycogen and fat. And then lastly, hydrate.
Oh,
No.
I'm not going to be able to yell at the kids when I get home.
Emergency.
I thought your kids were grown.
Grandkids?
Yelling consists of the fact that your rent is due.
Like that.
It is the only way I'm going to get them out of the house. I'm now to the point that I'm increasing the rent every month
Oh my God.
until they move up.
How old are they?
24 and 28.
Yeah, it's time. It's
It's
time.
expensive to live around
It's
here.
time to get
What's
out.
that?
It's expensive. I don't blame them.
I live at home, I'm 23, I don't care.
At 23, we didn't have a problem. But now they have
28.
jobs
Yeah.
and,
I can see the 28
And-
for sure.
yeah.
Jobs and lives and.
Well, we want them to have a life outside of the house.
Yeah. Why don't they get a place together?
Oh, because they kill each other.
Work opposite shifts.
Okay, let's go on. We are digressing greatly. Okay, your body stores energy. It stores it in fat. It stores it in glycogen. The glycogen in the muscles stays in the muscles. The glycogen in the liver, because of the enzyme, if I remembered, I would tell you, but you probably ought to know it. We'll get to it. There's an enzyme in the liver that breaks down glycogen that allows it to flow out into the bloodstream.
Tyrosine?
No, it's a glyco something.
Either way. keep it.
Glycosinogen? I like it.
Enzymes end with an ace, not a gin.
Glycosinolate?
Once in the blood, let's go on. Tissues have preferred energy sources. Brain glucose. 60% of your glucose goes to the brain. Brain does not store. It needs a constant supply. It does not have the ability to store glucose. Skeletal muscles at rest, fatty acids. Here we're back to the Krebs cycle, and I don't think you're going to need to know that. Adipose, fat. Adipocytes, fat cells.
Adipocytes and glucagon are the two fuel reserves.
Yeah.
How about adipose cells and lymphatic cells? That's what he said in class.
Adipocytes.
I know, I thought it was really weird.
They say lymphatic cells.
HEP.
Okay, liver.
Can you say that again? What did you say restores?
Liver and what was the other one? Adipose.
You have glycogen and you have adipose. That's how your energy is stored. Other than a little
And
bit.
the liver has glycogen.
The liver has glycogen and adipose. Yeah.
I actually have
Oh,
I really did not understand what is the primary way in which our bodies regulate whether to burn or build. It's a fairly broad question. A complete answer would take a while to compose.
Is it highlighted?
No, it's underneath the highlight, but I didn't even know where to start with this one.
Okay. What he's asking is, when do you store fat and when do you burn fat? To put it simply.
Burn and build.
Oh, remember we only use glucose for a certain amount of time, but we mostly use fat. Is that what he's saying?
Well, anabolic or catabolic. If you have a lot of calories, you're going to store fat.
Yeah.
If you don't have a lot of calories, you're going to burn the fat, and eventually you're going to burn glucose, then you're going to burn the fat, and then you're going to burn protein.
So anabolic is the one that you...
Anabolic is build. Catabolic is burn.
Insulin.
So it's all
You see what I'm saying?
Glucagon.
You know,
So it really depends on what is going on in your body at that moment in time.
yeah, it depends on your caloric intake and your caloric need.
Okay.
calories.
That makes
Yeah.
sense.
I mean, that's putting it very simply. But, you know, if you're taking in more, and we're talking about energy in that, if you're taking in more energy or more calories than you need, your body's going to, one, make sure there's enough glucose around, but that's not very much.
Right.
Two, it's going to be glycogen, and when that's kind of filled up, that's not a whole lot either. Three, it's going to go into fat.
So the more you eat, the more your blood sugar is going to go up, and then your insulin.
It's going to be pulled out, and the insulin's going to pull it out into the liver,
And then the less you eat, more glucagon
is going to be pushed out, and which raises the blood sugar.
Okay.
Okay?
Okay, that really helped. Thank you.
Got it? Go on. Energy is stored as white fat, as triglycerides. Hormones regulate it. Hormones break it down. Adipose cells secrete them that regulate hunger and metabolism. Now, adipose, that's all in the adipose cells. Now, we also have your Gavilan enzyme, which is a new enzyme they discovered in the stomach. It's in your book. But that is one that regulates your hunger pangs. That's one thing where having a vertical sleeve, having a Gavilan Y, gastric bypass, they take out that part of the stomach that produces Gavilan enzymes. I eat because it's a habit. I haven't broken it. But I've had a vertical sleeve. I eat because it's habit. I really don't remember when I walk in and say, I'm really hungry. I walk in and I say, it's 6 o'clock. It's time for a burrito. I walk in and say, I eat because it's more mental than anything else.
So you don't have the herpes?
Not really. But I still eat because I'm a notorious snacker. And that is probably why the sleeve didn't do me as good. I'm down 100 pounds. The sleeve didn't do me as good as a Gavilan Y might have, because I don't get sick when I eat.
But you can still get the lap band put on if you want to.
I'm not going to. I still can't eat over a cup. And I'm full. but then I can eat a cup in 30 minutes.
So
I'm
you're
sorry.
a grazer.
You have a question?
What if you messed it up? Instead of like having both the opposite of you, like keep feeding it instead of that, it's like not eating at a period of time.
I'm sorry, I didn't. What if you missed?
That ghrelin. So
Well, the ghrelin is, it just controls your hunger. And the more ghrelin, it kind of makes you feel hungry. So, I haven't heard of the negative side of it. Positive is it makes you hungry. I've got the negative side because they've taken that part of my stomach out and it's not hurting me. That's about all I
So
know.
have you reached a plateau?
I've stayed right at about 100 pounds down for the last 2.5 years. And if I eat, I go up a little bit and then I go down, and I go up and down. I really probably need to drop another 40 pounds. And I would feel real good and my wife would be off my case. That's a double bonus. Okay, white fat versus brown fat. I know you had that in here.
yes.
Brown fat is much smaller molecules than white fat. And it's burned, the proteins are released, you get a lot more heat released when brown fat is burned. That's why you see it in the babies, that's why it's really used in thermal regulation of the body temperature. Adults have enough other fat, enough surface area, whatever. They don't need it. Apparently we do still have a little bit of it. It's mostly in the children. Adipose development of
Skip.
adipose hormones.
What about the
Wait,
PPAR?
yes, that's there. Hold on.
PPAR
Are there? Yes. The back one. Hold on. It's in the.
He has it as a highlight. It's highlighted on the first page.
Yeah.
White and brown fat, adipokines, TNF alpha.
Yeah.
That was what you talked about, the UCP1 and the brown fat.
Yeah.
We did that in SI.
Yeah, it's in the chat.
Regulation of adipose tissues for fat, liver tissues for sugar, hormonal regulation of metabolites, insulin, glucagon, diabetes.
Yep.
I think we're there.
Yep, we're there.
Okay. Now the thing about the adipose sites, adipose cells, is if you're a child and you're in a real fatty diet, it creates more cells. As an adult, you don't lose them; they just get smaller. So the take-home message is: don't create them in the first place. No, the obese children are going to be obese adults, and they're going to have a lot more problems. So this pleasantly plump child is not doing the child any good.
The 100-pound 2-year-old.
Oh my gosh.
Been there, seen that. Not been there, but seen that.
Is that right?
Okay, adipose hormones, are we good?
Get going.
Skip that then.
Obesity.
He does talk obesity and diabetes.
Yes.
Okay, obesity. Risk factors for cardiovascular disease, diabetes, and cancers. As you put on more weight, you become more insulin resistant. The insulin resistance drives your hemoglobin A1C up. It drives your predisposition for type 2 diabetes. Not only are you insulin resistant, insulin resistant. The function of insulin doesn't function as well.
Is there a causal effect of the obesity and the diabetes, or is it just like associated?
It's causal. At some point, it moves into causal, because if you don't have the insulin, or if you're given insulin, then you're hungry and want to eat. There's more sugar. You got to take more insulin. Makes more fat, which lessens the effectiveness of the insulin, and it just becomes a wickedly bad circle.
Oh, because of the.
You see what I'm saying?
Yeah.
If you start on insulin, it lowers your blood sugar. You get hungry, your blood sugar goes up. You have to take more insulin. The more insulin pushes it into the fat. The fat makes the insulin work less well. It inhibits the insulin, and then you've got to take more insulin. and it just becomes into a really bad cycle.
That plays with the TNF alpha.
Right. It said that the more fat cells you have, the more TNF alpha you have.
And that's the factor that impairs the energy. Okay?
Okay.
Going on.
Yes.
Obesity. Hi.
Final here at 6 o'clock.
I know, and we will be out of here in just a couple of minutes.
Okay.
Okay?
Is that the teacher?
Yes, she is.
Okay, we're going to finish obesity and then we're going to take a break. Child is due to increasing number and size. In adult, it's in the size alone. If they're fat, if you feed them, if they get fat as a child, they're going to have more adipocytes. They're going to have more fat cells to deal with as an adult.
Wow.
In adult, they just get bigger or smaller. You don't lose them.
Never do something?
Never lose them unless you do liposuction.
But then again, they find a way around that too.
They do.
Yeah.
Yeah, but people have liposuction.
Body mass, BMI, rat studies,
Yeah, we don't need that.
regulation of hunger, digestive tract hormones, we've already been there, there's your ghrelin,
You better get out of here before she gets.
action of leptin, hunger regulation. Did we get into this? I don't think he has any of that on.
No, they're talking about thermal.
Oh wait, thyroxine, there it is.
Oh,
Thyroxine,
no.
thyroxine, lots of starvation response to
So we know about thyroxine.
Okay, let's take about 10 minutes and we'll meet back in 106.
106.
Okay?
Because I'm just gonna get there.